Hey, good morning or good afternoon, everyone. And in this lecture video, we'll be covering our drug metabolism. So, drug metabolism is one of the processes in which our body will remove the drug from our system. The elimination of most drugs from the body involves the processes of both metabolism, which will usually undergo first, then will be excreted by the kidneys. For many and most of our drugs, the principal site of metabolism is your liver. However, other tissues or organs may also have some associations with metabolism. So it's not limited to the liver, but the liver is the most highly important in terms of drug metabolism. So metabolism for short is a process of biotransformation or metabolism. It is the enzymatic conversion of a drug to a metabolite. So drug to metabolite is called our biotransformation or metabolism. And one of the key things or the key goals of our metabolism is to change the drug into a more polar form or water-soluble form. The reason being is that urine is water. Urine is mostly water, and the goal of the body is to make sure that the drug goes, or the metabolite of the drug goes to the urine and does not return back into systemic circulation. Hence, the goal is to make the drug more water soluble. So the liver is the major site of drug metabolism. There are a lot of reactions that undergo it. Examples like oxidation, reduction, hydrolysis, conjugation. These are the most common reactions, and we will be discussing them later between phase 1 and phase 2 reactions. So in terms of the drug biotransformation reactions, hepatic biotransformation is an important role in the inactivation and elimination of drugs. For most biotransformation, the metabolite of the drug is more polar than the parent compound, since the goal again is to make sure that it is soluble in the urine. The conversion of a drug to a more polar metabolite enables the drug to be eliminated more quickly than if the drug remained lipid soluble, since lipid soluble drugs tend to accumulate in the body, as we discussed in our distribution. Because lipid soluble drugs can cross cell membranes and can easily be reabsorbed, hence they tend to prolong in the body. In contrast, more polar metabolites tend to not accumulate and eventually go out into the urine. Okay. However, we do have some drugs that are exceptions to the rule, in which they are inactive when taken but become inactive once they are metabolized. We call these drugs prodrugs. So more and more research has been being placed to develop drugs which are intentionally prodrugs. The main reason being is so that pharmacokinetic profiles are better sometimes when they are prodrugs, meaning they are better if they are metabolized first, then afterwards they exhibit their systemic effects. Okay. In terms of physiology and anatomy of the liver is the major organ. It is both a synthesizing and excreting organ, and the basic anatomical unit of the liver is the lobule, in which it contains a network of interconnected lymph nodes and blood vessels, also containing your parenchymal cells. Okay. The liver is heavily perfused from the hepatic artery, and it has a large hepatic portal vein that collects blood from various segments of the GI tract. This one right here. So this is the hepatic portal vein. This is the largest vein, and most of the veins would meet in that location before heading to the liver. So the hepatic portal vein carries nutrients to the liver and accounts for 75% of the blood flow. Afterward, after the liver, the blood leaves the liver via the hepatic vein, which empties to the vena cava, which is part of your heart. Drug metabolism in the liver has shown to be both flow and site dependent, meaning the quantity of enzyme involved in metabolizing the drug is not uniform throughout the liver. Hence, if there are changes in blood flow, there is a chance that the drug would be it would take longer to metabolize, and it might need 2 or 3 passes. Clinically, hepatic disease such as cirrhosis can cause tissue fibrosis, and resulting changes of blood flow changes the bioavailability of drugs. So sometimes if you have conditions that affect your liver, it would affect bioavailability, meaning if you have liver damage, some drugs would actually increase their bioavailability since the first pass effect is less potent. Okay. We need to take into consideration the liver's condition when creating our pharmacokinetic models. And a good pharmacokinetic model should include the heterogeneity of the liver, the dynamics of blood flow, and the nonlinear kinetics of drug metabolism. Okay. Moving on to our hepatic enzymes. The liver is the major site of drug metabolism, and it is based on the reaction involved. The most common reactions is oxidation, reduction, hydrolysis, and conjugation, and they will be discussed under phase 1 and phase 2. The enzymes responsible for oxidation and reduction of drugs contain natural metabolites such as steroids are monooxygenase enzymes known as your mixed function oxidases. So your mixed function oxidases are one of the most important enzymes responsible for drug metabolism. The hepatic parenchymal cells contain these mixed function oxidases, and they are structural enzymes, which means they are already part of the liver, and they constitute an electron transport systems that requires reduced NADPH, molecular oxygen, cytochrome P450, NADPH, cytochrome P450 reductase, and also phospholipids. The phospholipid is involved in 2 ways. It binds the drug molecule to cytochrome P450, and it couples NADPH cytochrome P450 reductase to cytochrome P450. So it binds the drug molecule and NADPH cytochrome P450 reductase to cytochrome P450. So let's talk about cytochrome P450. This is one of the most important things in drug metabolism. So cytochrome P450 is a protein with an iron protoporphyrin 9 as the prosthetic group. Cytochrome P450 is the terminal component of an electron transfer chain, which I will show in the next slide, and it acts both as an oxygen and substrate binding locus for drugs, endogenous substrates in conjunction with the flavoprotein reductase and ADPH cytochrome P450 reductase. So many lipid-soluble drugs bind to cytochrome P450, resulting in oxidation or reduction. Cytochrome P450 consists of closely related isozymes, which technically are different components, but they are closely related in terms of structure. They have slight differences in their amino acid sequence, and they exhibit drug specificity. So these isozymes, which we will be discussing later, are specific for certain drugs. Okay, so here is your cytochrome P450 electron transfer chain. So in addition to the metabolism of drugs, your cytochrome P450 also catalyzes the biotransformation of endogenous compounds such as your steroids. And your CYP monooxygenase enzyme system is also located in other parts of the body such as your GI tract and your lungs. It's not limited to your liver. So the hepatic biotransformation enzymes play an important role in the inactivation and subsequent elimination of drugs that are not easily cleared through the kidneys. For these drugs, there is a direct relationship between the rate of drug metabolism and the elimination half-life of the drug. This means if the drug isn't cleared by the kidney as fast as the drug is being metabolized, the rate-limiting step for clearance in this step is metabolism. meaning the longer the metabolism takes, the longer the half-life of the drug is. And the biotransformation of drugs may be classified according to the pharmacologic activity of the metabolite or according to the mechanism for each biotransformation reaction. So here are the general biotransformation reactions and pharmacologic activity of the metabolite. So you have your active drug to inactive metabolite. Amphetamine undergoes deamination and becomes inactive phenylacetone. You also have your active drug to active metabolite, like your codeine, when demethylated, becomes morphine, which is actually more active than codeine. An example of an inactive drug to an active metabolite, this is your prodrugs. You have your hetacillin, when hydrolyzed, becomes ampicillin. And you have your active drug to reactive intermediate. When we say reactive intermediate, these are dangerous compounds since they can damage the body. Acetaminophen, when undergoing aromatic hydroxylation, gives off your reactive metabolite, which causes hepatic necrosis, your NAPK, N-acetyl benzoquinone. So going back to the pathways of drug metabolism, we have two main phases of drug metabolism. We have our phase 1 or our asynthetic reactions. Think of this as your unmasking. You remove parts of the structure. This includes your oxidation, your reduction, and your hydrolysis. Phase 2, on the other hand, is your synthetic reactions. This includes your conjugations. In the phase 2, this is when you attach more polar substances to the structure to make the whole drug structure more polar. In terms of which comes first, it's more common that phase 1 biotransformation occur first. They remove parts of the structure. Then phase 2 comes in and adds the rest of the polar components. So these reactions may include a high energy form of conjugating agent, such as your uridine diphosphoglucuronic acid, acetyl-CoA, 3-phosphoadenosine 5-phosphosulfate, S-adenosylmethionine, in which, with the appropriate transferase enzymes, combines with the drug to form the conjugate. Okay. In terms of the conjugation reactions, they may have limited capacity at high drug concentrations, which leads to your nonlinear drug metabolism. We discussed nonlinear drug metabolism back in our active transport systems, but let's review them. So when a system follows nonlinear kinetics, it means that when it becomes saturated, it reaches a maximum speed of the reaction. It usually looks like this. Okay. So take for example, this is the concentration. Okay. This is the concentration and this is your speed. The speed could be anything: speed of absorption, speed of distribution, speed of metabolism. But in this case, let's take speed of metabolism. So usually, when you have low concentrations, the speed is also low, since that's the maximum speed available. As the concentration goes up, the speed also goes up. However, when you have a nonlinear drug metabolism or nonlinear kinetics, there would come a point in which even if you add a certain concentration, the speed would not change. And this is what you call your Vmax. Okay, so the curve of speed versus concentration would look like this. There is a plateau. Okay, meaning there is a maximum speed. This is usually because a certain part of the system becomes saturated. It's like you're waiting for something to finish its process before you could load up the next part of the system. So this is just a review. We already discussed this prior. So glucuronidation reactions have a high capacity and may demonstrate nonlinear kinetics at very high drug concentrations. In contrast, glycine sulfate and glutathione conjugations have lesser capacity and demonstrate nonlinear kinetics at therapeutic drug concentrations. So these are things you need to consider when dosing. The limited capacity for conjugation pathways may be due to several factors. It could be due to the limited amount of the conjugate transferase. It could be the limited ability to synthesize the active nucleotide intermediate, or it could be due to the limited amount of the conjugating agent itself. So here are some common biotransformation reactions. And here are your phase 2 conjugation reactions. Please take note of the conjugation reaction, the conjugating agent, the high energy intermediate, and what functional groups are being combined. Please take note of this. This is also this would be asked. Okay. First, we have our acetylation. So acetylation reactions are important because the acetylated product is usually less polar. The acetylation of such drugs such as sulfanilamide, sulfadiazine, and sulfasoxazole produces metabolites that are less water-soluble and may cause sufficient concentrations may cause precipitation in kidney tubules, causing kidney damage and crystalluria. In addition, the N-acetylated conjugation reactions shows genetic polymorphism, meaning it's not the same for everybody. Some parts of the human population may not acetylate as other parts of the population. Take for example, African Americans and Caucasians are slow acetylators, meaning they acetylate slowly when compared to Asians. This is why some drugs are given in lower doses to the Americans while it is given in higher doses to Asians. This is not due to because of racism, but it's just the difference in our genetic makeup. Cases of this is your isoniazid, procainamide, sulfadrugs, and your dapsone. Next, you have your glutathione and mercapturic acid conjugation. Glutathione is a tripeptide of your glutamyl cysteine glycine that is involved in many important biochemical reactions. GSH is important in detoxification of reactive oxygen intermediates into non-reactive metabolites. And it is the main way we protect ourselves versus our electrophilic compounds. Through the nucleophilic sulfhydryl group of our cysteine residue, glutathione, GSH, reacts with non-enzymatically and enzymatically via the enzyme glutathione S transferase with reactive oxygen intermediates, particularly aromatic hydrocarbons formed during oxidative biotransformation. And the resulting GSH conjugates are a precursor of the drug conjugates known as your mercapturic acid or your N-acetylcysteine derivatives. Please take note that enzymatic formation of GSH is saturatable. This is the main way of why paracetamol toxicity occurs. If you have too many molecules of your paracetamol, it eventually depletes the GSH in the cells. This leads to reactive intermediate covalent compounds that destroy hepatic cellular macromolecules that leads to injury and necrosis. The suggested antidote for this overdosing is administration of your N-acetylcysteine, a drug that contains available sulfhydryl groups. So this is the chain of your paracetamol metabolism. You might be already familiar with it. So some peculiarities of our metabolism. So metabolism of enantiomers. So as you recall, enantiomers, these are the same compounds with slight differences within them. So many drugs are given as mixtures of stereoisomers. Each isomeric form may have different pharmacologic actions and different side effects. The rate of drug metabolism and the extent of protein binding are different for each stereoisomer. This means you need to consider enantiomeric forms when giving the drug into the system, since not all forms will exhibit the same propensity for metabolism. Similar to your stereoselectivity, you also have your regioselectivity. It means that hepatic enzymes can target specific parts of the drug molecule. A good example of this is your COMT or your catechol O-methyltransferase, in which it only metabolizes catechols. It doesn't and it only attaches the methyl group on a specific part of the catechol chain. Now, there are species differences in hepatic biotransformation enzymes. The biotransformation activity of hepatic enzymes can be affected by a variety of factors. In recent years, in vitro screening with human liver microsomes or hepatocytes have helped confirm whether a given CYP isozyme is important in human drug metabolisms. Okay. Next is we're going to delve into pharmacogenomics. So pharmacogenomics or pharmacogenomics is the study of the effect of different human genes in drug metabolism. What this means is, since we don't have all the same genes, some genes would affect drug metabolism differently than persons who don't have the gene. A good example of this is acetylation of isoniazid is genetically determined. If you are from the Americas, you are a slow acetylator, meaning isoniazid, you need to be given lower dosages of isoniazid. If you're from the Asia region, you are a fast acetylator. This means that you need to be given a higher amount of drug because you can metabolize the drug faster. This difference we refer to as genetic polymorphism. Another example of genetic differences is your G6PD deficiency, which is present in approximately 10% of African Americans. And another documented example with this enzyme was observed with phenytoin. There were two phenotypes, those who are efficient metabolizers and those who are poor metabolizers. The PM frequency in Caucasians was about 4%, and in the Japanese it was about 16%. Variation of the metabolic rate was also observed with mephobarbital. Okay. Next, we have our genetic variation of our cytochrome P450 enzymes. So the most important enzymes accounting for variation in phase 1 metabolism of drugs is the cytochrome P450 enzyme groups, which exists in many forms. Initially, cytochrome P450 enzymes were identified according to the substrate that was biotransformed. However, in more recent terms, the genes encoding many of these enzymes have been already identified. Multiforms of cytochrome P450 are now referred to as your isozymes. Okay, so when we say CYP, then a number and a letter, it is your isozymes. These are classified into families, denoted by Roman numerals, and subfamilies denoted by A, B, C, etc. Okay. Now, if an isozyme amino acid sequence is 60% similar or more, it is placed within the family. And within the family, isozymes of amino acid sequences of 70% or more are placed into a subfamily, and an Arabic number follows further classification. There is a new nomenclature that starts with CYP as the root, denoting the cyclopron P450. An Arabic number replaces the Roman numeral. So CYP, then a number, then usually there's a letter afterwards. So CYP or something. Okay. So take for example, CYP3A. So it means it's a cytochrome P450, 3 family, A subfamily. So it appears to be responsible for the metabolism of a large number of structurally diverse endogenous agents and synobiotics. CYP3A is one of the most popular. And in particular, CYP3A4 is one of the specific CYP enzymes which metabolize a lot of components. So an individual gene is denoted by an Arabic number after the subfamily. Take for example, you have cytochrome P450-1A2 family, subfamily, gene. CYP1A2 is involved in the oxidation of caffeine, and CYP2D6 is involved with the oxidation of drugs such as codeine, propranolol, and dextromethorphan. So let's review how to name a CYP and how to read a CYP enzyme. CYP meaning cytochrome P450. So the cytochrome P450 enzyme. 3, or it would be followed by a number. This is the family. Then a letter, subfamily, then another number, which is the exact gene. The well-known CYP2D6 is responsible for the brysoquine metabolism among individuals showing genetic polymorphism, and the CYP3 enzymes are known to be involved in the metabolism of vindesin, vinblastin, and other vinca alkaloids. Failing to recognize variation in drug clearance in cancer chemotherapy may result in greater toxicity or even therapeutic failures. Okay. Now we have our drug interactions showing drug metabolism. The enzymes involved in drug metabolism may be altered by the diet and co-administration of other drugs and chemicals. Enzyme induction is when a drug causes an increase in enzyme activity. So what happens is if you give this drug, it would stimulate the body to produce more of a certain enzyme. The opposite is enzyme inhibition. It is when a drug stimulates or reduces the production of a drug metabolizing enzyme. Both enzyme induction and enzyme inhibition is most applicable for our cytochrome P450 enzymes. And both of them can cause drug interactions. Okay, so here is a table of our common CYP inducers, or our enzyme inducers, and our enzyme inhibitors. Okay, so let's play a little bit of logic first. Okay. Take for example, we have a drug. So this drug could be anything. If you give if this drug is metabolized by an enzyme, and you give a drug an enzyme inhibitor. Okay. What happens is, since there is enzyme inhibition, the metabolism of the drug would slow down. If the metabolism of the drug slows down, the drug levels would increase. And since the drug levels increase, the therapeutic levels or the therapeutic effects also increases. However, it could lead to toxicity. Increased toxicity chance. If you give a drug with an enzyme inducer, since the enzyme induces more, since another drug can induce more of the enzyme, that enzyme would now metabolize more of the drug. It would lead to decreased drug levels, decreased therapeutic effects, and you shouldn't mind about toxicity. Now, the case is different if you give prodrugs. If you give it with an inhibitor, since the prodrug need to be metabolized before it becomes active, if it's not metabolized, it's not producing the active compound, meaning the active drug levels decreases. And the therapeutic effect also decreases. However, if you give it with an inducer, the drug levels increase since there is more of the enzyme that metabolizes the prodrug, meaning there's a faster conversion of prodrug to active drug. There is an increase in drug level. And there is also an increase in therapeutic effect. And there is also increase in toxicity chance. Okay, I hope that's clear. Drug with inhibitor, increased drug levels, increased therapeutic effects, increased toxicity. Drug with enzyme inducer, decreased drug levels, decreased therapeutic effects. The opposite is true for prodrugs. Prodrugs, when given with an inhibitor, decreases drug levels and decreases the therapeutic effect. Prodrugs given with an inducer increases the drug levels and increases the therapeutic effect and increases toxicity. I hope that is clear. Okay. Next, we have our autoinduction and time-dependent pharmacokinetics. So there are many drugs that enhance the activity of cytochrome P450 and change their own metabolism or the metabolism of other compounds. One good example of this is carbamazepine. Carbamazepine is what we call a suicide drug. The more you give carbamazepine, the more it induces the enzyme that metabolizes it. So in terms of outcomes, the longer you give carbamazepine, the less effective it is. Because since you give carbamazepine, which induces its own enzyme metabolism, if you give the same dose throughout, the less and less effective it would be. So when we say autoinduction, that's the case with carbamazepine. And when we say time-dependent pharmacokinetics, if you don't alter the dose, the longer you take carbamazepine, the less effective it would be. So to counteract this autoinduction and time-dependent pharmacokinetics, you simply need to increase the dose of carbamazepine as time goes on. You know what the first pass effect is? We discussed this in our absorption. And finally, we have our transporter-based drug-drug interactions. Examples include the inhibition or induction of transport proteins, such as your P-glycoprotein, organic anion transporter, organic anion transporting polypeptides, organic cation transporter, and your multi-drug resistant associated proteins. Here are some examples of transporter-based interactions. Okay, so that's the conclusion of our lecture. Please, I highly recommend that you read the learning materials provided and read the reference textbook. Again, our reference textbook is our Applied Biopharmaceutics and Pharmacokinetics. Okay, I hope you have learned something today, and let's see each other in the next video. Thank you.